Next, we will discuss uses of immunofluorescence, how and where we can use immunofluorescence. There are many uses of immunofluorescence. It is used to generate an image of organelles and cells with the help of using antibodies which are labeled with fluorophores, and these antibodies they target their antigens which are present in different organelles and cells. The specimen is visualized when there is binding of a fluorophore labeled antibody with its respective antigen present in the sample. So this gives its specificity that the antibody which is labeled with some fluorophore that binds the respective antigen only, it will not bind to other places. And this is achieved due to absorption of radiations of specific wavelength by the fluorophore and then its excitation. We have already discussed that when a particular fluorophore is used, it absorbs specific radiations, then it gets excited and energy released that is used to see that particular antigen. It's a technique to detect the distribution of proteins or other molecules of interest in the given sample. Many fluorescent stains are designed for different biological molecules. We have discussed that we can use different types of fluorophores, and specific fluorophores they target their respective antigens or tissues in the cell in the sample. For example, this DAPI that is specific for adenine and thymine regions in nucleic acids in living or fixed cells, and when we say they are targeted by these, it means they are excited by particular wavelength, here in this case by UV light. So DAPI is specifically used for nucleic acids staining. Similarly, phyllidine is used to visualize actin filaments in living or fixed cells or in vitro studies. जब हमने सेल्स का माइक्रोस्केलेटन जिसमें एक्टिन फिलामेंट्स मायोसिन फिलामेंट्स जो हैं वो टारगेट करने हो तो हम फ्लाइडीन जो है उसको यूज करते हैं. Similarly, fluorochromes such as fluorescein are chemically linked with specific proteins or other structures in the cells. So ज़ाहिर है along with DAPI, phyllidine, this fluorescein that is also used, and that is able to stain specific proteins on the cells. Other types of dyes like Alexa-Fluor dyes, they're also used to stain cells and tissues. These Alexa-Fluor dyes, they are very beneficial if we talk about other fluorophores. The reason is that other fluorophores like phyllidine, like fluorescein, FITC and RITC and others, they get photo-bleached with the longer exposure, while Alexa-Fluor dyes, there is less problem of photo-bleaching. So that gives advantage to Alexa-Fluor dyes as compared with other dyes. Similarly, they give more strong signals as compared with other dyes as well. So different fluorophores are used for different samples, for different types of samples. DAPI is used for nucleic acids, phyllidine is used for actin filaments, fluorochromes like fluorescein that is used for staining different specific proteins. Alexa-Fluor dyes, they can also be used.